Hi everybody. Today we shall discuss some of the questions from one of the important chapters for NEET, that is photosynthesis in higher plants. From this chapter, one or two or three questions can be asked, which are mainly based on its mechanism and concepts. Let us see some of the critical questions from this particular chapter. Coming to the first question, photosynthesis is a physico-chemical process. The physical process of photosynthesis is movement of electrons from reaction center to primary electron acceptor. No, this is chemical process, not physical process. Transfer of light energy in light harvesting complex of photosystems. Yes, from one pigment molecule to another pigment molecule, the transfer of absorbed light energy is a physical process. It takes place by inductive resonance or resonance transfer. Photolysis of water, it is a chemical process. Formation of ATP by chemiosmosis, chemical process. So, if the enzyme is involved in the reaction or process, or electrons are either ejected or accepted, it is a chemical process. So, in the second one, it is a physical process because light energy is transferred from one pigment molecule to other pigment molecule in a light harvesting complex or antenna complex by inductive resonance. It is a physical process. Second option must be the correct one. Let us see which is given. Second one is given. Here it is a solution. Transfer of light energy among the pigments of light harvesting complex and to reaction center is physical process of photosynthesis as the energy is transferred by inductive resonance. Movement of electrons from reaction center to primary electron acceptor of either PS1 or PS2 is a photochemical process as there is expulsion or an acceptance of electrons. Photolysis of water to photochemical process as the light energy is used to pull the electrons from water. ATP formation in light reaction of photosynthesis is a chemical process as there is phosphoanhydride bond formation by the activity of ATP synthase. Coming to the next question. In which of the following experiments related to necessity of a factor for photosynthesis, potassium hydroxide is used? So it is used to demonstrate in an experiment where CO2 is necessary for photosynthesis. So that experiment is Moll's half-leaf experiment. One option must be the correct one. So let us see the answer. Third one is given. Solution. In Moll's half-leaf experiment, cotton dipped in potassium hydroxide solution is kept in the test tube to create carbon dioxide for environment to show the necessity of carbon dioxide in photosynthesis. Variegated leaf experiment shows photosynthesis only in the green or chlorophyllous parts of plant. Black paper strip method demonstrates necessity of light in photosynthesis. Hydril law funnel experiment demonstrates the release of oxygen in the presence of light in photosynthesis. So third option must be the correct one. Coming to the next question. Two statements are given. Let us see the statements. The alignment of chloroplasts of a green cell perpendicular to the incidence of light is called diastrophy. Absolutely true. And this diastrophy alignment of the chloroplast is seen in low and moderate and optimum light intensity. So statement 1 is correct. Statement 2. Chloroplasts show diastrophy in low and optimum intensity of light. This is also correct. Both the statements are correct. So fourth option must be the correct one, which says both statements 1 and 2 are correct. Let us see. Fourth one is given. Solution. Chloroplasts are aligned along walls of green cells to obtain optimum quantity of light. Alignment of chloroplasts perpendicular to the incidence of light in low and optimum intensity of light is called diastrophy or epistrophy. Alignment of chloroplast parallel to the incidence of light in highlight intensity is called parastrophy. This is parallel to the incidence of light. So, fourth option must be the correct one. This explanation is also given clearly. Coming to the next question. Correct statement you have to choose. Chlorophyll A can act as accessory pigment as well as reaction center. Yes, chlorophyll A is present in the light harvesting complex as well as reaction center in core complex. The first option must be the correct one. Why 2, 3 and 4 are wrong? Chlorophyll B can act as accessory pigment as well as reaction center. No, not reaction center, only accessory pigment. Chlorophyll A always behaves as reaction center. No, can also behave as accessory pigment. Chlorophyll B shows more light absorption peaks than chlorophyll A in red region. No, chlorophyll A absorbs more red light than B. Coming to the answer. Which must be the answer? Chlorophyll A can act as accessory pigment as well as reaction center. That is the correct answer. 1 is the solution. Chlorophyll A acts as accessory pigment when present in light harvesting complex and as reaction center in core complex of photosystems. The remaining 3 also we can read out. Chlorophyll B is always an accessory pigment but never be reaction center. Chlorophyll A not only behaves as reaction center pigment but also behaves as accessory pigment. Chlorophyll B shows less red light absorbency than chlorophyll A. Assertion reason. Given below are 2 statements. One is labeled as assertion A. So it is assertion reason type question. Let us see the statements. Photosystem 1 transfers electrons to PS2 during photosynthetic light reaction. Never. Assertion is wrong. Electrons are always transferred from PS2 to PS1, never from PS1 to PS2. Assertion is wrong. Oxidized reaction center of PS2 is reduced by electrons obtained from PS1. No. Oxidized reaction center of PS2, that is P680, is reduced by getting electrons from water, not from PS1. Assertion is wrong. Reason is wrong. So the third option must be the correct one. Let us see the answer. Electrons are transferred from PS2 to PS1 during photosynthetic electron transport. Oxidized reaction center of PS2 is reduced by electrons obtained from water liberated in photolysis of water, but not by getting electrons from PS1. Next one. It is matching type. Answer is 2. Let us see whether answer 2 is correct or not. Role of air in plant growth. Joseph Priestley. A4. A4. Liberation of oxygen in the presence of light from plant green parts. Engine house. B3. See, formation of glucose in green parts of the plant. Julius Van Sachs. B. First action spectrum of photosynthesis. That is given by Engelmann. So A4, B3, C2, D1, that is the second option must be the correct one. Role of air in plant growth, Joseph Priestley. Liberation of oxygen in the presence of light from green parts of the plant, John Engine House. Formation of glucose in green parts of plant, Julius Van Sachs. First action spectrum of photosynthesis was given by T.W. Engelmann. Coming to the next question. A, B, C, D, E, 3 statements are given. Let us see which statements are correct and which statements are wrong, but we have to identify the correct statements. Cornelius Van Neel showed that CO2 is reduced by hydrogen obtained from oxidizable compound. Correct statement as given in the book. H2S is oxidized in photosynthesis of green and purple sulfur bacteria. Given in the book, it is also correct. While molecules of water are used for the production of a glucose molecule in photosynthesis, it is also clearly given in the equation as given in the book. Right? Water is not only involved but also released in photosynthesis of air plant. Yes, 12 molecules of water is involved, 6 molecules of water is liberated for the formation of 1 glucose molecule by reducing 6 CO2 molecules. E. Light reaction occurs in stroma of chloroplast. No, it takes place in grana or granathylakoids or thylakoids of chloroplast. A, B, C, D are correct. E is wrong. So third option must be the correct one. Let us see the answer. Answer 3. Solution. Cornelius Van Mill showed that CO2 is reduced by hydrogen obtained from oxidizable compound. H2S is oxidized in photosynthesis of green and purple sulfur bacteria. 12 molecules of water are used for the production of a glucose molecule in photosynthesis. Water is not only involved but also released in photosynthesis of higher plants. Light reaction occurs in thylakoids of chloroplasts but not stroma. Eighth question. Choose the correct statement. Correct one you have to choose. Carotenoids, chlorophyll A and chlorophyll B are present in light harvesting complex. Absolutely true. This is the correct statement. P700 is a part of antenna complex of photosystems. No. It is the reaction center of PS1. P680 is the reaction center of PS1. No. It is the reaction center of PS2. Movement of electrons from primary electron acceptor of PS2 to P700 is uphill movement, no downhill movement. So, first option must be the correct one. Here is the solution. Carotenoids, chlorophyll A and chlorophyll B are present in light harvesting complex. P700 is reaction center of photosystem 1. P680 is the reaction center of PS2. Movement of electrons from primary electron acceptor of PS2 to P700 is downhill movement. Choose the incorrect statement. Incorrect one you have to choose. Photolysis of water is associated with PS2. True. Because oxygen evolving complex is attached to PS2 towards the lumen side of the thylakoid membrane or granathylakoid membrane. Right? So this is the correct statement, but we want incorrect statement. The electrons removed from PS1 are replaced by PS2. Absolutely true. The protons and oxygen formed due to splitting of water released into lumen of thylakoid. It is also true. So fourth one must be the incorrect one. When PS2 is operational, the process called cyclic photophosphorylation takes place now. When PS1 is operational and PS2 is not operated, cyclic photophosphorylation takes place. So fourth one is the incorrect one. Let us see the answer. Fourth one. Here is the solution. Cyclic photophosphorylation, only PS1 is operational but not PS2. Oxygen evolving complex and PS2 are associated with each other in granathylakoid membrane and both together involved in photolysis of water. Oxygen and protons are released into lumen of thylakoid and electrons are carried through PS2, PS1 and ultimately to NADP plus to form NADPH. Then only light of wavelength beyond 680 nanometers is provided to a plant or a potted plant. Only PS1 is operated and cyclic electron transport or photophosphorylation takes place. Let us see the options. What happens when light whose wavelength is more than 680 nanometers is given? And actually PS2 is not operated. Only PS1 is operated. Both the cyclic and non-cyclic electron transport takes place. No, only cyclic. Only non-cyclic electron transport is operational. No, only cyclic. Cyclic electron transport takes place only in stromal MLA but not in granal MLA. No. PS1 is present in both granal MLA, stromal MLA. Hence, in both grana and stroma thylakoids, cyclic electron transport takes place. 3 is also not correct. Cyclic electron transport takes place in both granal MLA and stromal MLA. This is true. Fourth option is the correct one. Let us see the answer. This is the explanation. If light of wavelength beyond 680 nanometers is provided to a plant, it is absorbed by the pigments of only PS1. Hence, only cyclic electron transport takes place. Since PS1 is present in both grana and stromal MLA, cyclic electron transport takes place in both types of MLA. 11th one. Given below are two statements. You have to identify which statements are correct, which statements are wrong. During photosynthetic light reaction, the concentration of protons is more in lumen than stroma of chloroplast. The protons of the lumen is more than stroma. Absolutely true. High accumulation of protons in lumen is only due to release of protons in photolysis of water. No, not only due to release of protons due to photolysis of water into lumen, but also pumping of more and more protons from stroma to lumen through PQ cycle. So statement 1 is correct, statement 2 is incorrect. Statement 1 is correct, but statement 2 is incorrect. First option is correct. Here is the explanation. Accumulation of more protons in the lumen during light reaction is not only due to Photolysis of water, but also pumping of more protons from stroma to lumen through proton pump, in which plastoquinone and cytochrome B6 subcomplex are involved. Assertion reason type. During light reaction of photosynthesis, high proton gradient is established between stroma and lumen across thylakoid membrane. Absolutely true. Protons of stroma are not only pumped into lumen but also used for the formation of NADPH from NADP plus in light reaction. Absolutely true. A is correct, R is correct. Reason clearly explains the assertion. Hence, A and R are correct, and R is the correct explanation of A. The fourth option is correct. Let us see. Yes. The reasons you can find out A, B, C reasons are good. Pumping of protons from stroma to lumen due to operation of PQ cycle. Usage of protons of stroma for the formation of NADPH. This is also reason for establishment of proton gradient. Release of protons into lumen due to photolysis of water. All the three reasons are correct for establishment of lot of proton gradient across the thylakoid membrane between stroma and lumen. Next one. The first found product of biosynthetic phase of photosynthesis due to uptake of CO2 was demonstrated by Melvin Calvin in algae, very clearly given in the book. It demonstrated this one in green algae, chlorella and Senidesmus by using C14, carbon 14, carbon labeled carbon dioxide with 14 isotope, radioactive isotope. He demonstrated. He also used the techniques of paper chromatography and autoradiography. So algae is correct. Let us see. Yes, fourth one. Here is the solution. Melvin Calvin for the first time showed in green algae, chlorella and Senidesmus, the first found product of biosynthetic phase due to uptake of carbon dioxide as 3-carbon compound 3-phosphoglyceric acid. Coming to the next question. Matching. Answer these three. Here it is. The primary CO2 acceptor. What? List 1, list 2 with respect to biosynthetic phase of photosynthesis in maize. This you have to match. Primary CO2 acceptor is phosphoenol pyruvic acid. A2. Where is given A2? Third and fourth options. Primary CO2 acceptor, phosphoenol pyruvic acid is present in mesophyll cell chloroplasts. Mesophyll cells. Right? A2. First product of primary carboxylation oxaloacetic acid. A2, B4. So third option must be the correct one. A2, B4. Secondary CO2 acceptor, secondary carbon dioxide acceptor. That is ribulose bisphosphate 1, C1. The first product of secondary carboxylation. That is 3-phosphoglyceric acid or phosphoglyceric acid 3. Third option must be the correct one. Let us see the answer. Third one. Here is the solution. Primary CO2 acceptor phosphoenol pyruvate. Secondary CO2 acceptor RUBP. First product of primary carboxylation oxaloacetic acid. First product of secondary carboxylation 3-phosphoglyceric acid. Third option is correct. Coming to the next question. For the formation of one glucose molecule through Calvin cycle, how many ATP are used in reduction stage? In Calvin cycle, for the formation of one glucose, 18 ATP, 12 NADPH are used. Out of 18 ATP, 12 ATP are used in reduction stage and 6 ATP in regeneration stage. Neither ATP nor NADPH are used in carboxylation stage. Right? So answer must be the 2 here. Right? Let us see which answer is given. 2. Here is the explanation. For the formation of one glucose molecule through Calvin cycle, 6 carbon dioxide must be reduced to form a molecule of glucose. For this, Calvin cycle is operated 6 times. Each CO2 reduction, there is usage of 2 ATP in reduction stage. For reduction of 6 CO2, there must be usage of 6 into 2, that is 12 ATP in a reduction stage. Identify the correct statements. You have to identify the correct statements. In Calvin cycle, usage of ATP is more than usage of NADPH. Absolutely true. 18 ATP are used, 12 NADPH are used. A is the correct statement. In Calvin cycle, usage of NADPH is more than number of CO2. Yes. For 6 CO2, 12 NADPH are used, or 1 CO2, 2 NADPH are used. B is also correct. In carboxylation stage of Calvin cycle, only NADPH are used. No, nothing, neither ATP nor NADPH are used in carboxylation stage of Calvin cycle. C is wrong. B, in reduction stage of Calvin cycle, only ATP are used. No, not only ATP but also NADPH are used. In the regeneration stage of Calvin cycle, both ATP and NADPH are used. Wrong. Only A and B are correct. First option is correct. Let us see which is the correct one. First one is correct. It is explanation. In Calvin cycle, for the formation of a glucose molecule, there must be carboxylation of 6CO2 and utilization of 12 NADPH and 18 ATP. Neither ATP nor NADPH are used in carboxylation stage. Equal number of ATP, that is 12, and NADPH are used in reduction stage. Only 6 ATP are used in generation stage. From this, very clear about understanding or concluding which statements are correct, which statements are wrong. Option answer 1 is correct. Means A and B statements are correct. Coming to the next one. The name transanatomy to the leaves is given by the observing the morphology or nature of. The transanatomy is given based on the morphology of bundle sheath of C4 plants like maize and sorghum, like even sugarcane. Bundle sheath cells of C3 plants do not have chloroplasts. Bundle sheath cells of leaves of C4 plants have got chloroplasts. Hence, the bundle sheath with chloroplasts appears as a green garland or wreath-like structure around the vascular bundle. The name transanatomy is based on mesophyll. No, not mesophyll. Epidermis, not epidermis. Bundle sheath, yes. Distribution of vascular bundles, no. It is morphology of the nature of the bundle sheath. Answer 3 must be the correct one. Let us see the answer. Answer 3. Solution. The name transanatomy is given due to the ring or garland-like colored appearance of bundle sheath around the vascular bundles. Coming to the next one. Choose the correct statement. You have to choose the correct statement. C4 pathway has one carboxylation and one decarboxylation reactions. Right. It has one carboxylation and one, no, it has got two carboxylation and one decarboxylation reaction. First carboxylation takes place in mesophyll cells or mesophyll cell chloroplasts. Second carboxylation and decarboxylation, only decarboxylation, takes place in bundle sheath cells. So, first option is wrong. C4 pathway has a two carboxylation and one decarboxylation reaction. It is true. This is a correct statement. Two carboxylation, one in mesophyll cells, another one bundle sheath cells. One decarboxylation in bundle sheath cells. It is correct. Second option is the correct one. Third one. C4 pathway has only two carboxylation reactions, but not decarboxylation reactions. Yes, it has got decarboxylation reaction too. Third option is wrong. C4 pathway has one carboxylation and two decarboxylation. Wrong. It has one decarboxylation, two carboxylation reactions. So the second option must be the correct one. Second one is given. Here is the answer. Now the solution. C4 cycle has the two carboxylation reactions, that is RUB carboxylation in bundle sheath cells, PEP carboxylation in mesophyll cells, and one decarboxylation, malic acid decarboxylation reaction which occurs in bundle sheath cells. Coming to the next one. Choose the incorrect statement. In photorespiration, Rubisco catalyzes oxygenation reaction. Absolutely true. But it is correct statement, not incorrect statement. But we have to identify incorrect statement. Photorespiration is more common in C3 and less common in absent in C4 plants. It is the correct statement. Right? We have to identify incorrect one. In photorespiration, oxygen is utilized and CO2 is released. This is also true. Oxygen is used in peroxisomes and chloroplasts. CO2 is released in mitochondria. So naturally the fourth one must be the incorrect one. In photorespiration, NADPH are not formed, but there is formation of ATP. No. ATP is also not formed. Neither NADPH nor ATP are formed. Moreover, ATP are utilized in chloroplast for the formation of phosphoglyceric acid from glyceric acid. The fourth option must be the incorrect option. Let us see the answer. Here is the solution. In photorespiration, neither NADPH nor ATP are formed, but ATP are used for the phosphorylation of glyceric acid by glycerate kinase in chloroplasts. Coming to the next question. Which is the most common limiting factor for photosynthesis? Most common limiting factor and most important limiting factor is carbon dioxide, because the CO2 concentration in the atmosphere is less than optimum concentration. Hence, it is usually the limiting factor. Light is not the limiting factor. Light intensity most of the times is more than required in nature, or more than optimal light intensity is available. Temperature more than required is available in many cases. Water, usually it is not deficient, usually sufficient amount of water is present. Usually in dry conditions only, water deficiency noticed and it may behave as limiting factor. But here CO2 is the most common limiting factor. Second option must be the correct one. Let us see. Second one is given. CO2 is the major limiting factor of photosynthesis. Light behaves as a limiting factor of photosynthesis in plants growing in shades and in cloudy days. Temperature and water seldom behave as limiting factors of photosynthesis in nature. Like, comment, subscribe, share, and hit the bell icon to get the notifications as soon as they are published. Thank you very much.